All right. So, like I said, we're starting today with Unit 4. Oh, Mother Jones, it's not casting. Mother Jones, where did my Chrome go? Sorry, those of you watching. Yeah.
I'm not. I'm not. I'm trying to do it.
You got it. You mean like roast
Sorry, those of you at home. It is trying to start up. We lost a connection. All right, am I casting?
No. Oh yeah. No you are.
All right. So, Unit 4 is energy. All right. And so that's going to encompass everything from enzymes to cellular respiration to photosynthesis to trophic levels. So, having to do with producers, primary consumers, secondary consumers, all of that stuff. So, we're going to go over all of that today. We're going to start with enzymes. All right. And so that's where we're going to start today. Enzymes. They have one big function. So the main function of an enzyme is to lower something. Anyone remember what it lowers?
The reaction.
Lowers activation.
Energy.
All right. And so it's going to speed. So in turn, by doing that, it speeds up reactions. I'm going to put RxNS. That's an abbreviation for that. Activation energy, if you ever see it on an energy diagram, you might see it abbreviated as E of A. So capital E of, and then capital A. That's energy of activation. Enzymes speed it up. All right, so they catalyze. That's what you'll hear that term a lot also, that enzymes catalyze a reaction, meaning they lower the activation energy and in turn speed it up. I should probably close that parentheses. Okay? And so if I look at just simple vocab and pictures and stuff of it, a normal enzyme, if I look at it just with a basic diagram, it's going to look like this. And then I'm going to also have this. And then I'm going to have this. Okay? This thing right here, my Pac-Man looking guy, is going to be called my enzyme. This right here, the thing that's going to plug into the enzyme, is called my substrate. And the place where it plugs in at is right here. The place where it plugs in at is called an active site. All right. And then finally, when that substrate binds in that active site with the enzyme, it's going to catalyze this reaction and it's going to create our products. And that's what this is the product. Okay. I see a lot of you escribing, so get a haircut. You look good.
I think I look decent. That's what my girlfriend told me.
Okay. Michael, you can put that on the ground if you want. Can't depend on others to do everything for you, Michaela.
Yeah.
I'm not spicy today, and you are. Yeah, you are. You just got mad because you weren't even mad.
All right, so lowers the activation energy, speeds up reaction. So enzyme substrate plugs into the active site, and it's going to make the product. Okay, let's look at these energy diagrams if I were to look at this. So let's say I've got this right here. This would be my energy. This would be time. So if I were to look at a diagram, it would probably look something like this. And then I could have another line that goes just like that. So if I were to make a little key, black is going to be without enzyme. Red is going to be with enzyme. Okay? And so I could label these. There's a couple labels we're going to look at. So from this point on the graph to where it started with, this is what's known as the E of A, or activation energy without enzyme. And this one right here, from here to here, is the E of A with enzyme. What do you notice about them? If I were to compare those two. Okay, which one goes up less? The one with an enzyme is a lot less. All right, it doesn't have to climb anywhere near as high. And remember what we said the main function of an enzyme was? To lower activation energy. That's what this diagram shows you. If I add an enzyme, it lowers activation energy, thus in turn speeding that reaction up. And there's one last thing that we need to have labeled on this thing that you might see. From here, so where I started with to where I end at is known as something. It's known as delta G. The change in free energy that I have, all right, is known as delta G. I could either give energy off or I could take energy in. And that's going to depend on which kind of a reaction it is. And we're going to talk about those two in just a second once you finish writing this. So those are the three big ones that you need to look at. So it's from where we start at to where we end at. Not taking into account everything in the middle is going to be my delta G. Any questions thus far? All right. So now we're going to look at these two diagrams. So if I look at this one, and I say it goes like that, and I also look at this one, and it goes like that, okay? So I'm looking at both of these. The one on the left, did it give energy off or did it take energy in? Look at the end compared to the beginning. Do I have less than I started with or more? I have less. If you have less of something, did you give something away or did you take it in? I gave it away, so this one right here is going to be exergonic. The one on the right, I have more than I started with, so I took it in, meaning it's going to be endergonic. All right. And then remember, change in free energy. If I took energy in, it's going to be positive delta G. If I gave energy off, it's going to be negative delta G. This gave it off, so this is going to be negative delta G. This took it in, so it's going to be positive delta G. Okay. And we've got some other terms. If I'm taking, or if I'm giving energy off, am I building something or am I breaking something apart? I'm breaking it apart. So we have two terms. We have catabolic and anabolic. Which one of those means to break down? Catabolic. So this would be a catabolic reaction, meaning this one right here is an anabolic reaction. All right? And if it's catabolic, or let's say anabolic, I consider anabolic reactions comparable to or analogous to cleaning your bedroom. Does your bedroom spontaneously clean itself? No. I wish. It doesn't. So anabolic is going to be not spontaneous. Meaning that catabolic is going to be spontaneous. Your room definitely doesn't spontaneously clean itself up, but it seems like it spontaneously got messy. All right? So exergonic, negative delta G, catabolic, not spontaneous. If I'm comparing these two between cellular respiration and photosynthesis, one of them builds something up, one of them breaks something down. Which one of them breaks something down? Cellular respiration breaks down glucose. So this diagram right here on the left, does it sound like a horse outside? cellular respiration. And this one right here that's taking energy in, positive delta G, anabolic because I'm building something or synthesizing something, is photosynthesis. Photosynthesis. Okay. Any questions on this?
Why did you write those two in different colors?
Just because it's an example of a process that does that.
Okay.
So this would be like if I were to give you a diagram and say, which does this diagram represent photosynthesis or cellular respiration? This one represents cellular respiration. But cellular respiration isn't the only thing that could look like that. Okay. Cool. You guys ready for some cell resp? We're going to go over these and draw these. As we go over these, I'm also going to talk about what some of the biggest areas were that people missed on their exams. Okay. Anyone else need any of that? None? So, oh. Okay, cellular respiration. I've got to start with something. What's the big molecule that I want to break down? C6H12O6, also known as glucose. And I have to have oxygen, 6 of them. And they're going to combine to make 6 CO2 plus 6 H2O. Okay? And ATP. And so I've got to undergo these 3 big steps. Step number 1, the very first one that does not require oxygen, so it's considered anaerobic, is going to be glycolysis. And so I'm going to start with this 6 carbon right here, and it's going to go to make these 2 3 carbon molecules. Do you remember what the 3 carbon molecules are called? Pyruvate. Pyruvate. Okay? So I've got these pyruvates. To get this thing started, though, it's going to require a little energy. So the very, very first part of glycolysis is endergonic. So right here, something's got to come in. And it's going to be 2 ATP come in and 2 ADP come off. So I've got to input a little energy. Okay? And in doing so, it's going to split this thing. And on both sides now, something comes in, something comes off. NAD plus comes in. And the same thing happens on this side. NAD plus comes in. Remember what comes off? NADH. NADH comes off. NADH comes off. All right? And it's beautiful. But that's not it. But wait, there's more. Also, 2 ADP plus an inorganic phosphate come in on both sides. 2 ADP plus an inorganic phosphate come in on both sides, and 2 ATP come off. Oh, that was janky. Just like, yep. 2 ATP come off. And so the name of this step, we said, is called glycolysis. Glycolysis. And it happens in the cytoplasm. Okay. One of the most common questions you'll see about this is it's going to ask at the end of glycolysis, where's the most amount of energy stored at in which of these molecules? And if you were to see it on a multiple guess test, like the AP exam, it'd say, is the majority of the energy stored in the ATP produced, the NADPH produced, or is it stored in that pyruvate? The most amount of that energy. The majority of the energy is stored in the pyruvate. And the reason why is because if you think right here, I only make 2 NADHs. I only make a net total of 2 ATP. But in the overall process, I make a crap load more of NADHs and a crap load more of ATP. And it's because of everything that's still stored in that pyruvate. I just have to unlock it during the next step called the citric acid cycle. Okay? You guys still need this? Do what? Cytosol, cytoplasm, synonymous. Tomato, tomato. Okay. So, oh. All right. Now I'm going to zoom out so I can go to my next step. That's the outer membrane. Okay. So, along the way, after I made one of these pyruvates, I'm not done with it, and I say, hey, it's time to go on a trip in our favorite rocket ship. And in doing that, it's going to form a molecule called acetyl, ACETYL-CoA. All right, or a 2-carbon molecule, which is only, somehow went from a 3-carbon to a 2-carbon. Okay, it lost a carbon, and that carbon gets given off in the form of what? It gets given off as the form of CO2. And remember, anytime CO2 gets given off, NAD plus comes in, and NADH comes off. Okay, so I made some NADHs here. I made a CO2. This is, we have a name for this step. It's considered a sub-step of the citric acid cycle. And the name of this step, we call it, you remember? Ooh. Oxidation of pyruvate. And it happens on the intermembrane space. Of the mitochondria. Okay. So Demetria just told us this is called oxidation of pyruvate. No, I'm proud of you for knowing that. Okay, that's, in my opinion, one of the harder ones to know. But I'm talking, we threw out this nerd word, oxidation. And it's like, hey, yo, slow down there with that vocab. So we're going to look at this acronym real quick. I don't know if it's an acronym or whatever they're called. Oil rig. And oil rig stands for something. Oxidation is losing. Reduction is gaining. Okay. So this says, as Demetria just told me, oxidation of pyruvate. That meant pyruvate lost something. It lost an electron, and it lose that electron attached to a hydrogen. And I know that because NAD plus came in, and what did NAD plus pick up? It picked up a hydrogen. The only place it could have gotten it from is from that pyruvate that got fed into this cycle. Okay, so pyruvate got fed in, pyruvate loses a hydrogen and an electron, thus why it was oxidized. All right, and that means if pyruvate was oxidized, NAD plus gained an electron, so NAD plus was what? Starts with an R, ends with reduced. It was reduced. Okay. Okay. And so now we're left with this molecule called acetyl-CoA. It's going to combine with something. It's going to combine with a 4-carbon molecule. You don't need to know the names of those. If you really want to, it's called oxaloacetate, I think. And it forms this 6-carbon molecule called citric acid. And that goes to a 5-carbon molecule. And that goes to a 4-carbon molecule. Whoa. And that goes to a 4. And that goes to a 4. This is a janky one. I know. And we made this little cycle. And this cycle is called the citric acid cycle, or Krebs. And its location is the matrix of the mitochondria. Okay. So I'll let you get this written down, and then we will start drawing what comes off of this.
freaking bottom, dude.
All right, ready? Okay. I was told no. Are we okay now? Okay. So, if I go from a 6 carbon, whoa. If I go from a 6 carbon to a 5 carbon, I lost a carbon. And remember, carbon gets given off as the form of what? CO2. And anytime a CO2 comes off, NAD plus comes in and gains an electron. So NAD plus comes in and gets reduced to form NADH. Same thing happens here. CO2 gets given off and NAD plus comes in
Can't see the top, so.
and gets reduced to form NADH. Can't see the top? All right, and then now I'm at 4 carbon to 4 carbon. On this one, we didn't lose a carbon, so what actually happens here is a DP plus inorganic phosphate comes in and forms a TP. Oh, I mean put 2. Got a little excited there. Right here, another thing happens. FAD comes in and forms FADH2. So this FADH2 and these NADHs, remember we said they have a purpose. It's to carry something. They need to carry electrons, and they're going to carry it to the oxidative phosphorylation step, otherwise known as electron transport chain. So that's their whole thing, is to carry these hydrogens with these electrons on them. And then finally, right here, from this 4 to 4, it goes NAD plus comes in and it gets reduced to form NADH. And all is well on the Western Front. And that's our step right here. So if we were to look at it as according to a lot of textbooks, as of right here where that green line is, starts the citric acid cycle. So if it were to ask you, during cellular respiration, where is CO2 produced? And you might see it labeled as like step 1 is glycolysis, and it'll label it step 1, step 2, step 3. Glycolysis being step 1, citric acid cycle step 2, and oxidative phosphorylation step 3. And it might ask you, identify which steps CO2 is produced. And it'll say step 1 and 2, step 2 and 3, step 1 and 3, step 2 by itself, or step 1 by itself. All CO2 produced during cellular respiration comes from citric acid cycle alone. Okay? None of it's produced during glycolysis. None of it's produced during oxidative phosphorylation. All of the CO2 we make is produced during the citric acid cycle. ATP is produced during all 3 steps. NADH is produced during steps 1 and 2. FADH2 is produced during the citric acid cycle. Okay? And the whole purpose why we don't make any of those NADHs or FADH2s during the citric acid cycle is because that's where they go to get broken down. And that's what we're going to go to right now. Okay, so let's zoom in. This step is called oxidative phosphorylation. And it happens in the inner membrane of the mitochondria. All right, and we've got to look at this now. So if I draw this, I've got an integral protein, peripheral proteins, like these transmembrane proteins. Yeah, everything's cool right there. And then I've got this big molecule right here. Okay, so right here, something's going to get brought in, all right, to get oxidized. Remember, if something gets oxidized, is it gaining or losing? It's losing. So something's going to come in and lose an electron right here. That thing that comes in is NADH comes in. And it's going to lose an electron forming NAD+. And right here, FADH2 comes in and FAD gets given off. Right? So both those things lost an electron, therefore they were oxidized. Thus where oxidation comes from in oxidative phosphorylation. Okay? When that happens, remember, I ripped off hydrogens from both of them. They're going to get pumped across this membrane. So hydrogen goes across, and now I end up building up this big proton gradient. So I've got H+, 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 all these H pluses over here. And it doesn't like that. Too many of these, doesn't like this thing. And so it's going to flow down this gradient. And it's going to flow through this molecule right here. Do you remember what this enzyme is called? It's called ATP synthase. And so it's going to flow from high to low, because remember, high is on the outside. It's going to flow from that way down through this thing, and it's going to function kind of like a water wheel. So remember, we've got this water wheel, and it's going to turn it. And as it turns it, it generates energy. It generates enough energy to take ADP plus inorganic phosphate. And remember, we said we have this fancy term to add a phosphate onto something. Do you remember what that term is? Phosphorylation. It's going to phosphorylate ADP to form ATP. Interesting. NADH came in and was oxidized, meaning it lost an electron, which generated this huge concentration gradient of these hydrogens, which then went through ATP synthase, turning it like a little water wheel. And remember, when water turns a little water wheel, it generates energy. And when it did that, it generated enough energy to phosphorylate ADP to form ATP, or oxidative phosphorylation. We couldn't phosphorylate ADP without oxidizing that NADH. We couldn't oxidize NADH if we didn't reduce it earlier in the step by breaking down glucose. So the whole purpose of this is to generate these NADHs and these FADH2s. That way they can come to this electron transport chain. That way they can be oxidized. That way we can phosphorylate ADP to make ATP. And we're left with these last things in the end where finally I've got two H pluses plus 1/2 of an O2. And if I combine those, what do I get? I get water. And that's what my final electron acceptor is at the end. That was for the your Unit 4 exam. For the cellular respiration part of oxygen, that was the biggest thing that people forgot at the end, was saying that oxygen accepts those hydrogens at the very end to form water. Okay, so they just said hydrogen goes through ATP synthase, down this concentration gradient, blah, blah, blah. But they never said what finally accepts it at the end. Okay? Any questions before we do photosynthesis? Yup. Hey, how you doing? All right. Yeah. So, that's everything. Did you?
From college.
Nice.
The box. I'm gonna burn it on Saturday.
Are you? Why burn it?
Huh?
Why burn it?
I wanna keep it.
No.
Anyway, you had 6th grade prep, right?
Yeah.
Do you want to take my class Friday at 6th grade? To be able to?
This coming Friday? Yeah, I can.
I'll let Leslie know if that's cool.
Yeah, I probably can. Yeah. That works. Yeah, let me know.
Sure now. It's Monday. What happens between here and Friday? The art, 6th period.
I most likely can. If I can't, I'll just tell her to get someone else. It's fine.
There probably is nobody else. That's the problem.
Yeah. I have no problem with it. I'll do it.
Cool.
All right, thanks.
I'll let her know then.
Okay.
Thank you.
Yep.
I'll see you in the morning.
Later.
You're lying.
Okay. And so now we've got to do photosynthesis. We've got a little bit more left for photosynthesis. Or we've got to start photosynthesis. And our first step on there is, if I look at photosynthesis, remember I start with 6 CO2 plus 6 H2O gives us C6 H12O6 plus 6O2. Okay. So I have to have CO2 and water, and I combine those to make glucose and oxygen. I realize my feet just keep getting closer and closer to your space, because now I'm like sitting like this.
Oh, I can't say what I want to say.
Okay. So first step for this is called the light-dependent reaction. Okay, in this light-dependent reaction, it's going to happen on the thylakoid membrane. And looks like this. And I've got a couple things on there. So the very first thing that I've got to have sunlight. Because, remember, this reaction is light-dependent, meaning it must have light. Okay? Who would have thought that that's what it meant? And then I've got these things right here. I've got these little molecules. I've got a couple of them. There's two that I'm going to color green. Okay. And I've got one more I'm going to put right here. Okay. This first one that I have is called Photosystem 2. And it likes a wavelength called P680. This one's called Photosystem 1. And it likes the wavelength P700. I colored these green. Okay? What does green have to do with a plant? It's the color that it does not like. It's the color that chlorophyll gives it because that is the wavelength of light that it is reflecting. The plant does not absorb the color green. It reflects the color green. That is why you see it as green. Okay?
So it's everything but green.
It is absorbing all the other colors but green, and that's why it looks like that. And this other one is called cytochrome. Cytochrome. C-Y-T-O-C-H-R-O-M-E.
Something dramatic so you can get a sick day this week.
I have 42 of them. If I wanted a sick day, I'd just take one.
What is that? 70?
P700.
42 sick days?
Yeah. Okay. So, if I look, the sunlight spits off energy. And that energy is going to be used for something. That energy is going to split a molecule. Do you remember which molecule it splits?
Electron.
It's going to split water to generate electrons. So it's going to split water and generate 2H plus and 1/2 of an O2 molecule. This is one of the biggest things people missed on the oxygen one also. They didn't say that, because remember how you had to do cellular respiration and then to photosynthesis for the oxygen? They didn't say that oxygen was generated during photosynthesis because sunlight comes in and splits a water molecule. They may have just said, well, water gets split and it makes hydrogen and oxygen, but you didn't say how. What splits it? Sunlight splits it. Okay? And then the next thing they missed is they never said what happens to the oxygen. Okay? This O2 molecule, waste. It exits the cell. So it's going to be a byproduct. As long as you said that, like, sunlight breaks down a water molecule and it's going to split it into hydrogen and oxygen, and then from there the oxygen is going to leave. That's fine. Okay? So your verbiage may have been a little bit different. Okay? When it did this, it split water, and what also happened is an electron was removed. That's what he was just telling us. And that electron's going to get excited at the photosystems, and then it's going to travel down, and then it's going to hit another photosystem, and it's going to get excited, all right? And it's there. Electron made that little pathway. And I've got one last molecule that's not on here. Nope, that one's upside down.
What happens when you assume?
It's okay. It doesn't matter. Okay. The name of this molecule, ATP synthase. Okay? When I split this, I generated a lot of H pluses. H plus, H plus, H plus, H plus.
What?
Okay? I generated all these hydrogens right here. Just like earlier, it doesn't like that. And so these H pluses are going to flow through this. And they're going to go right here. Okay? Same thing, same type of molecule. It's like a water wheel, and it's going to turn. And it's going to take ADP plus an inorganic phosphate, and it's going to phosphorylate it into ATP. This one's called something different, though. This isn't oxidative phosphorylation. This one uses the sun to phosphorylate it. So, do anyone remember what this step's called? Or this process right here is called? Photophosphorylation. Okay. So, ADP to ATP. And then finally, there's one other thing I've got to produce here. Right here, remember that electron? I generated these hydrogens over here, and I got this electron. Something's going to come in and collect that electron and hydrogen. NADP plus H plus is going to come in, and NADPH comes off. So at the end of this, I produce this, and I produce this. I produce NADPH, and I produce ATP. And they have one goal, or one purpose. Their purpose is to drive the Calvin cycle, because that's what our next step is. This was the light-dependent reaction. The Calvin cycle is also known as the light-independent reaction. Yes? H plus. Yes?
So what does it do when the hydrogen flows to the ATP synthase?
It turns the ATP synthase kind of like a water wheel. And when it flows through it, by turning that little water wheel, it generates enough energy to phosphorylate ADP into ATP. So we need ATP, and so that's why we've got to split water, is to make that proton gradient, or all those H pluses on the outside. That way it'll go through ATP synthase, that way it'll generate enough energy to make ATP, because we have to have ATP to drive the Calvin cycle. Okay? And so, which leads us in? Calvin cycle. Or light independent. Reaction. I'm not gonna lie, every time I spell independent, I sing that stupid song.
Good song.
Every time. It's the same thing with the Bananas song.
This beat is bananas. B-A-N-A-N-A-N.
Yes. All right. So, Calvin cycle, or light independent reaction, meaning the Calvin cycle can happen whether it's dark or light out. I start with 3 RUBPs. Ruby Ps. And do you remember what happens to these Ruby Ps?
They go back.
Whoa. These Ruby Ps form 6 3-PGAs. But the only way they can do that is if something gets entered. 3 CO2s. On the Unit 4 exam, this was the absolute most common missed thing on there that people didn't say. They never said that CO2 comes in and it binds with Ruby P. Okay? It's called fixation. That's what the name of this step is. It's the most commonly missed thing out of all of it. If I look, a good amount of you had it. There was, yeah, about a third of the people who did theirs didn't put that step on there. Okay? It's on yours. Yours on the campus of Infinite. Okay? So, it forms 6 of these 3-PGA molecules. And those 6 3-PGAs are going to get rearranged to form... Hey, guys, cut the talking, please. Okay, to form 6 G3Ps. And in doing that... Okay? I've got to have 6 ATP come in and 6 ADP come up. Where did those ATP come from? They came from the light-dependent reaction. Very good. Thank you. Okay? Because if you look in there, normally we don't associate ATP being produced during photosynthesis, and the reason why is because all the ATP we do produce gets broken down in the Calvin cycle. And so, not only did I need ATP, I also have to have 6 NADPH come in and 6 NADP plus come off. So NADP plus, or NADPH, lost an electron, so it was oxidized, meaning those 3 PGA molecules were what? They were reduced. NADPH was oxidized, meaning those 3 PGA molecules were reduced. Okay? And now I'm left with 6 of these G3Ps. And what's going to happen is those 6 are going to go to 5. 5 G3Ps. Meaning I lost a G3P. And one G3P is essentially 1/2 of a what? Of a glucose, yep. Okay? And I don't like those G3Ps, and so what's going to happen is I'm going to add a little energy to rearrange it, to reform this Ruby P. And I'm going to add 3 ATP come in, and 3 ADP come off. And now I've got names of these steps. This first step is called fixation. This second step is called reduction. And this third step is called regeneration. Because Ruby P has carbon dioxide fixed to it, or fixation occurs where I fix CO2 onto Ruby P. 3 PGA is reduced because the NADPH comes in, gets oxidized, drops off a hydrogen, reducing it to form those 6 G3Ps. 1 G3P gets given off, and now I'm left with 5 G3Ps. I don't like these 5 3-carbon G3Ps, and so I'm going to regenerate Ruby P. So I'm going to take those 5 3-carbon G3Ps, switch it around, and regenerate 3 of those 5-carbon Ruby Ps. Thus where the name regeneration comes from. Okay, because it's just about rearranging stuff. Okay? Any questions on any of these 3 things? Okay? There's one last thing we're going to talk about, ecosystems real quick, and then we're done. It's going to take like 2 minutes.
Yeah, do we need a new page or like
No, I mean, I'm literally just going over trophic levels like primary consumer, secondary consumer, tertiary consumer. Okay? So, come on guys, hang on there, a couple more minutes. Trophic levels. Trophic means energy. So we're talking about energy levels in ecosystems. The most basic one that makes its own energy is called a producer. Okay? So it's a producer. They're called autotrophs. Because they make their own energy. Auto means self, energy or troph means energy. And so producers are their own autotrophs. It's mainly going to be stuff like plants. Okay? Then I go to my next thing. My next level up is going to be primary consumer. Okay? That primary consumer only eats plants. And we have a term for a thing that only eats plants. What's that called? Herbivores. Okay? So it's an herbivore. And so this is going to be like a lot of insects, a lot of just plant-eating animals like deer, cows, horses, stuff like that. Then we can have a secondary consumer. Okay? That secondary consumer, it can eat the primary or it can eat the producer. So they can either be what's called carnivorous. What does carnivorous mean? Only eats meat. Only eats meat. All right? And so it can either be carnivorous, or we have another term for something that eats both, meat and plants, or omnivorous. So it can either be a carnivore or an omnivore, meaning it eats both. Okay? And then finally, we have a tertiary. It can be both as well. Generally, tertiaries tend to be mainly carnivorous, but it can be both. Okay? For instance, us. All of these three right here, if I were to group these three, do these things make their own energy or do they get it from other stuff? Do we know a term that means different or other? Heterotro. Because these things don't make their own energy, they get their energy from eating other organisms. Okay? And then we have one last one called a decomposer. Eats dead stuff. Worms, some insects, stuff like that. Okay? So it's going to eat dead or decaying organisms. And that's a wrap, folks.